All right, so recording has started, I think. Hello, everyone. This is our great pleasure to have today Kate Lynch. Kate is a colleague based in Melbourne, Australia. She's a philosopher of science. She works on causation, explanation, and causal reasoning in biology and medicine with many different topics where she applies these very important questions. Um, she recently was very much involved in discussions with the lay public and the media as well. This is something rare, but I think very, very important. Few philosophers of science do that, and she's one. And recently, her work has been featured on ABC News, different radios, the Los Angeles Times, I think, and also she received a prize, which is a media prize this year, the Australian Association of Philosophy Media Prize. I also want to say that for the Fit in Biomed network, having strong connections with Australia has always been crucial. Paul Griffith retired recently, as you all know, and now spends quite a lot of time in the UK, which is great. But simultaneously, we want to keep some strong connection with Australia, and Kate is the future, with others, of course, from this point of view. I really want to thank you, Kate, for making yourself available at 6 p.m. Australian time. And I very much, as everyone else, look forward to hearing you on human genetic causal reasoning. The floor is yours, Kate.
Thank you so much, Tamara. And thank you for the kind introduction. I do want to clarify that with that media prize, I shared the prize with Christopher Lane, who's also a philosopher of biology, just so that I'm not scooping all the credit there. We did a project together on de-extinction. But I'm going to talk about something totally different today. So this is sort of more my expertise, talking about genetics, causation in genetics. And in particular today, I'm going to talk about causal reasoning, so the way that people tend to perceive different kinds of causal relationships. I'm going to start off just framing these questions in terms of what philosophers are interested in when they think about causation. So, in case there's sort of people here without a strong philosophical background or don't know much about the philosophy of causation, the kind of traditional things that philosophers are interested in when they think about causation, and this is, there's a long history of thinking about causation in this way, is giving some sort of definition or account to tell us what exactly causation is. So if you've got two things, whether they're events or variables or objects, this C and E here on the screen, we're trying to say, what exactly does this arrow mean? What feature of the world are we picking out when we're looking at causal relationships? What is causation? These are sort of big general problems in philosophy of science, as well as metaphysics and other areas of philosophy. And related to this question, an account of causation should be able to identify should be able to tell us which are the causal relationships and distinguish these from other kinds of relationships where we sometimes get similar results, where things are associated. So, for instance, we want an account of causation that tells us about this arrow between C and E, but is not going to capture some sort of relationship that we might see, like a correlation between the two E variables here on the screen. So the answer to one, what is causation, should give us at least an account of how we capture this question too. But something I think that has received less attention and is getting a bit more increasing attention now is this third question. And that's when there's multiple different causes for a given effect, which there almost always is. This is areas of complex causation. How do we decide which causes we pick out as more salient, more important, or a better causal explanation compared to others? So when you've got lots of different causes for an effect, how do we decide which is the cause or the most important cause or the kind of cause we should focus on when we're trying to give a causal explanation? And a classic example of this from the philosophy literature is you might ask the question, okay, what caused the candle to light? Was it the striking of the match? Or was it the presence of oxygen in the room? And the intuition here is supposed to be that the striking of the match is the important cause in this scenario, and the presence of oxygen in the room, where it's causal in some sense, we relegate that to a mere background condition. It's not importantly causal in the same way as the striking of the matches. So when you have multiple different things that are necessary to produce an effect, you get this problem of how to pick out one. Why do we say that the striking of the match is more important compared to something like oxygen? And we get this causal selection problem as well when you start going back in time along a causal chain. So in cases where we think causation is transitive, we might go back and say, well, why did I strike the match? Well, I struck the match because there was a power outage, say. Why was there a power outage? Because there was a storm. What caused the storm? Maybe it's because we're in the end of a La Nina cycle down here. And as we go back in time, we can see one thing causing another. Yet we don't want to go all the way back that causal chain when we try to explain why the candle lit or even why the match was struck, right? There's a point in a causal chain where people tend to stop and focus on the cause or the causal explanation. Similarly, with oxygen in the room, you can go back in time here and talk about the great oxidation event or cyanobacteria being responsible for the oxygen in the room, but that doesn't seem like a relevant causal explanation. Not many people would point to that and pick that out as the cause of the thing that we're interested in, the candle lighting. And so there is a little bit of a literature, both in philosophy and in psychology, about what are the kinds of features, both of causal relationships and of cause and effect variables themselves, that lend themselves to picking out causes as more important or as better explanations for an effective interest. So when it comes to features of the causal relationship, this is this arrow, this causal relation connecting C and E. There's been a few features proposed that either justify or at least explain why some things seem more important than others when we talk about causation. I'm only going to have time to talk about two of these today, which is specificity and invariant, one version of invariant stability. And I know some people on the Zoom call have done quite a lot of work about all the different ways that you can flesh out these concepts. I'm going to be talking about just one version of that. But there are others as well. So proximity, in the example I gave before, the more proximal causes seem to me the more appropriate to causal explanations than the very distal ones. And you might think there's pragmatic reasons for picking out causes as important as well, like how easy they are to intervene or whether we can actually go into the world and do something about those causes, manipulate them. And so there's been a little bit of literature just in philosophy talking about how these things explain causal selection, or in some cases justify it, give good reasons to say that this cause is more important than this cause. There's also features, and you might think just a little flash there, that's the limits of my PowerPoint animation skills, but features of the actual causal variables themselves, the cause and the effect, which are called causal relata. And there are some features of the causal relata which also tend to lend themselves to the way that people causally reason when they think that some causes are more important than others. I'm not going to focus too much on these in this talk, but we can maybe get into it in discussion because I think these things are relevant in the genetics cases. But these include things like whether the cause is perceived as abnormal, the moral valence, the perceived moral valence of the cause and the effect, whether we think these things are good or bad, and also whether there's a presence of a human agent in the causal system. So our causal attributions are quite tightly linked to our moral attributions. And it looks like when we think there's a morally responsible agent, often a human agent, then that's going to influence the way that we causally reason about things. So I'm going to move on to just talking about these two features of the causal relation, specificity and invariance, and then show how they bear out in the genetics cases. So first of all, specificity. There's lots of different ways of conceptualizing specificity in biological systems. I'm just going to be talking about one kind, which is a sort of one-to-one mapping between different causes and different effects. And a kind of intuitive way of getting a handle on what specificity is thinking about biological specificity, say between enzymes and substrates, where you have this lock and key model, where there's a particular enzyme that matches up with a particular kind of substrate. There's an example from James Woodward in a non-biological example where he talks about the turning of a radio dial as a specific cause of the radio station that you get to listen to. Again, this is a little bit of a dated example, I guess now maybe that the radios don't work in quite the way you see here on screen, but each little turn of the dial corresponds to a very specific output, the effect output being a different radio station. And this is in contrast to a non-specific cause of what we're going to listen to on the radio, which might just be the on-off switch. Right, so the on-off switch is still important. We still need the radio to turn on so that we can listen to that station, but it doesn't give us any kind of fine-grained control like a specific cause would, like the turning of a radio dial. And you can see on the screen here a kind of visualization of what this one-to-one causal specificity looks like, where you've got a cause producing just one kind of an effect, and that effect being realized by just one kind of cause. We've seen this kind of idea come up in epidemiology. So the Bradford-Hill criteria actually refers to specificity in a couple of the different indications for causality. But the one most relevant here is the condition that an exposure is related with the outcome in question and no others, and the outcome is associated with the exposure in question and no others. So it's got this sort of one-to-one relation. The Bradford-Hill criteria aren't strictly criteria for causation, but they're supposed to lend evidence to whether there's a cause and effect relationship out there in the world. And so according to this criteria, if there's this one-to-one specificity, there's good reasons to think there's causation. And the important thing to note about specificity is it's not an all or nothing feature of a causal relationship. You don't have just specific and non-specific causes, but it's a matter of degree. So you can have more specific causal relationships and less specific causal relationships as well. So that's specificity. Another feature of causal relations that seem to be important when we pick out causal explanations or more important causes is how invariant or how stable that causal relationship holds up across changes in context or changes in background conditions. So again, I'm going to see how my PowerPoint animation skills go to demonstrate this one. What I've got here is two causal relationships on screen, C1 and E1 and C2 and E2. And some background conditions are going to flash up, which are going to just be different colors. And you'll see that as these background conditions change, the colors change. The causal relationship on the left, C1 to E1, is maintained. It persists across those changes in background conditions. Whereas the causal relationship on the right disappears at some point and then reappears. So it only persists over a more limited range of background conditions. And that's also represented by this little box down the bottom here, where you can see those black lines represent the causal relationship persisting, and the colours in the background represent the contextual or causal background change. So philosophers like James Woodward have said that invariant relationships are valued by ordinary people and by scientists, and that they're justified in valuing these kinds of relationships. So not only does this explain why we pick out different kinds of causal relationships as more important or more valuable, but it gives us good reason to pick these things out. It justifies that selection. And another philosopher of science, Sandra Mitchell, has written quite extensively about how this feature of causal relationships might lend itself to a theory about what scientific laws are, or at least one feature of scientific laws, where a scientific law would be maximally invariant. It should hold across all kinds of different contexts, perhaps every context across the universe, across different scales and across space and time. So that's what we might call a scientific law, whereas something that's more fragile, that only holds up under certain background conditions, we're not likely to grant it that kind of law-like status. And again, like with specificity, this notion of invariance is one of degree. You can have things that are more or less invariant or more or less stable across background conditions. As well, in epidemiology, I mentioned the Bradford-Hill criteria with specificity. They also have something like this when discussing consistency, and we see this parallels with stability in other areas of medical research as well, like the discussion of external validity in randomized controlled trials. That also kind of relates to stability. Okay, so how does this apply to genetic causation, which is my interest? Well, in order to answer that question, we first have to specify exactly what the causal relationship is when I'm talking about genetic causation. Because at a very proximate level, we can talk about DNA causing RNA sequences. Now you can imagine that would be characteristically specific. This is something we think is a real feature of the DNA-RNA relationship. Similarly, DNA to amino acids is a very specific causal relationship. But what I'm interested in is the much more distal causal relationship between DNA and a trait. So, for instance, a DNA sequence and the ability to roll your tongue, which you can see here on the screen. And the reason I'm interested in this is the bulk of the discussion in the areas I'm interested in behavior genetics. And also, this is the kinds of causal relationships that people tend to entertain when they're thinking about genetic causation in the public realm, which is also something I'm quite interested in. So as well as the public communication of philosophy of science, I'm trying to understand what's going on with the public and collect data from the public to understand their causal reasoning. And so if we want to understand the causal relationship between DNA and a particular trait, again, I'm going to relativize this to just human populations, then we still need to specify further exactly what that causal relationship looks like, because there's different kinds of ways that genes can cause traits or trait differences. One thing to distinguish, and this is for all sorts of causal relationships, is that you can have token causal relationships and type causal relationships. Um, so, for instance, if we stick to the tongue rolling example, I can roll my tongue. So we might make a claim, if I'm making a claim about myself, that I've got a particular gene, and that gene causes my tongue rolling ability. So I'm making a token causal claim. It's about a single instantiation of the causal relationship between DNA and that particular trait as borne out in one individual person. You can also make type causal claims about the same kind of causal relationship where we say it's not just me, but in general, at a population level, this gene, gene X, causes tongue rolling. And this is if we're identifying just a single gene as the cause of the trait of interest. Now, there are plenty of traits where we do try to identify just a single gene or a few combinations of genes to explain a trait or to explain some sort of variation in a trait. But a lot of genetic explanations, particularly in behavior genetics, which is more my area of research, are based on quantitative analysis, such as heritability estimates, where we're looking at traits that are caused by the action of many, many, many genes, as well as often having environmental components. And in this case, what we're actually trying to explain is the variation in that trait within a population. And if you see these kinds of claims in the press and even sometimes in academic texts, you'll see claims like my tongue rolling is mostly due to genes. So people give some sort of quantitative estimate. This will sometimes correspond to heritability estimates, which will be like it has a heritability of 0.8, or people will say 80% genetic, which is a bit of a problematic way of interpreting those results. But it's important to note that with those studies, with the quantitative analysis, it doesn't make sense to talk about genetic causation at an individual level in the way that the single gene trait does. At a type level, you might be able to make a claim saying tongue rolling is mostly genetic, and I'll explain sort of how that works in heritability in a moment. But one hopefully way to get an intuitive grasp on why this doesn't work in the individual case is, say we have a heritability estimate for something like height, which is roughly 80%, 0.8 heritability. So we might say height is mostly genetic. If we look at an individual like myself, so I'm 165 centimeters tall, it doesn't make sense to say that 80% of my height is genes, right? I can't count up 132 centimeters. They're the genetic ones. And then these ones up here, they're due to the environment. It just doesn't make sense to partition things in a percentage, using a percentage when it comes to the individual level. This is something we can only do when we're looking at variation in a population. So that's why this little text in the box here is in red. We can't make token causal claims about using heritability estimates. There's quite a lot of debate as to whether we can even make type causal claims using heritability estimates. And I'll get to that a bit later on. So even when we're talking about this DNA to trait relationship, there's lots of different ways you might be able to talk about causation in this context. Okay, so let's talk about single gene traits first. I think these are a little bit simpler to understand, and these, to remind you, are ones where we can make token and type causal claims. And so single gene traits are the sort of traits you would have learnt about in school when you learnt about genetics. Mendelian traits is another way of thinking about them, or monogenic traits. And one thing about these single gene or Mendelian traits is that they do tend to be characteristically specific. So they very often have the relationship where it's one gene variant, one mutation in one gene, producing one kind of outcome, quite often a disease outcome. There are some cases, such as this example here, the outcome here is osteogenesis imperfecta, which is a disease that causes brittle bones and a whole host of other symptoms, where there's two genes that if you have a mutation in either one of these, you'll end up with osteogenesis imperfecta. So it's not completely specific, but it's relatively specific if you have a mutation in either one of these, and these are on completely different chromosomes, then you'll end up with this disease. However, specificity, the way that we assess specificity can in part depend on how the variable is described. So if we're describing the outcome variable as a disease, then yes, it looks quite specific. If we describe the outcome, though, in terms of their symptoms, you can see that there's less specificity. There's less of this one-to-one mapping between a particular kind of cause, the gene or gene mutations, and particular effects. So it's important to note that these features like specificity are dependent in part on how things are described. And you might have other good reasons to justify why you describe a disease as this um, amalgamated set of symptoms. Now, as well as being characteristically specific, single gene traits, Mendelian traits, are often quite invariant, and this lends people towards deterministic thinking when they think about single gene traits. There are, of course, counterexamples. So the classic one is phenylketonuria, which if you have a gene mutation for phenylketonuria, you will present with the disease, but exactly how that disease presents depends quite heavily on the environment, on the kind of diet that you have. So you can modify your disease presentation quite significantly if you have phenylketonuria by subscribing to a particular kind of diet. This is variation in expressivity, and that this can depend on people's environmental backgrounds as well as their genetic backgrounds. Another example of things not being deterministic or not being completely stable or invariant is incomplete penetrance, where different gene mutations will sometimes present with the outcome, quite often a disease, and sometimes not. And that will be modified based on other kinds of background conditions, people's genetic backgrounds or their environmental backgrounds that they're in. But a lot of single gene monogenetic traits are quite invariant. So, for instance, blood group, this is a non-disease example. It doesn't really matter what your diet is or what your other genetic background is, your blood group will be quite highly determined by whatever gene variant you have in that Mendelian sense. And the thing to note about single gene traits is the gene in these cases are very often or almost always picked out as the cause of the disease or of the trait. And this is a clear case of causal selection when people pick out this gene, this single gene mutation, as the cause of the outcome. And I think that's related to them having these characteristics like specificity and invariance. It's also worth noting, though, that monogenic traits or Mendelian traits almost always relate to disease outcomes. So this database here, the Online Mendelian Inheritance in Men, estimates that 97.6% of the traits in that catalogue are diseases. And diseases by their nature are characteristically abnormal and negatively valenced. And these are also things that feature in the way people causally reason when they pick out particular causes. So that might also be having an effect. Okay, so that's single gene traits. They're characteristically specific, they're characteristically invariant, which lends towards a bit of deterministic thinking about genes. And as people like Gregory Raddick have suggested, because this is the way we learn about genetics in school, because this is the way people tend to think about genetics from an early age, this might be influencing the way people causally reason about genetics in other cases, even when this specificity and invariance is perhaps not present. And there's these other cases that are captured in quantitative genetics, quite often by this metric heritability. So heritability concerns the causes, or at least the explanations of variation in a trait, where you have a lot of continuous variation in the way a trait is expressed. So again, sticking to this example of height, you can see this nice distribution here of these men differing in height, where most of them are around average height. Some are taller, some are shorter. And you might want to ask the question, okay, we see a lot of variation here in height. How much of that variation is due to genes? And how much of that variation is due to these young men having different environments? And in the case of height, people think it's mostly due to genes. That the heritability estimate for height is about 80%. So 0.8. And so we say most of the variation in populations of height is due to variation in genes. And it's the effect of many, many genes, the very small effects of many, many genes. And in fact, a lot of the time we still can't capture that at a molecular level. This is the problem with missing heritability. But it's the effect of many, many genes kind of abstracted here on this graph on the left, as well as interactions in the environment that produce this variation in populations. Now, exactly how to interpret the heritability statistics, something like height has a heritability of 0.8, is still subject to a lot of contention, and this is particularly in the case of behaviour genetics, where people are looking at the causes of things like academic attainment, intelligence, psychological traits, personality traits, aggression, criminality. All of these things have had heritabilities estimated for them, and there's this real spectrum as to how people causally interpret a heritability estimate. So right down the non-causal end of the spectrum, and this is a paper, this is a picture taken from Adele and Hardin's 2022 BBS paper. You have people like Richard Lewontin and Ned Block who think that heritability is a totally useless concept. It doesn't give us any causal information at all. It's almost in the realm of pseudoscience. And then you have right up the other end of the spectrum, the other extreme are people like Arthur Jensen saying that if we get the heritability of IQ, this is in the context of looking at IQ and intelligence, then this reflects some sort of innate, genetically determined aspect of intellectual ability. So in this case, the heritability estimate is not just telling us about genetic causation, it's telling us about genetic determinism, a very strong sense of genetic causation corresponding to this sort of stability and invariance concept. So there's this whole spectrum as to how to interpret heritability estimates. So this is a debate in behavior science and in behavior genetics and in philosophy of science about how to interpret heritability at all, whether it's causal at all. Is it something that we should just dismiss like phrenology, or is it something we should take seriously? And one of the reasons people tend to doubt the utility of heritability estimates, so right down the Lewontin end, is that they are concerned about how well you can extrapolate the results from one study to other populations. Right, so this is a quote here from Richard Lewontin from a very influential paper criticizing the heritability estimate, saying that heritability only applies to the sets of environments in which that determination was made, in which that estimate was made, and it can't be extrapolated to other distributions or other environments. So you might do a study with those men on the grandstand and you get a heritability of 0.8, but all that tells you is about the variance in that population. We can't make a claim about height in general in other populations. That's the criticism. Now, you might say, okay, well, what if we study lots and lots of young men, and we look at heritability of height, and we see that even across different kinds of environments, we're getting a similar kind of heritability estimate, and that is what's happening. Oh, sorry, I'm just getting a pop-up. I hope my computer's not going to do anything silly. See how that goes. Right, so you might have corresponding to what we've got down the bottom here, we've got two different environments. E1 and E2, these could correspond to two different studies where you're looking at the heritability of height. And we're seeing, it's just simplified here to three different genotypes. We're seeing similar kinds of things, a similar kind of outcome on the y-axis across these different environments. But the criticism still remains from people like Lewontin who think, well, what about the untested environments? What about the future environments in which nutrition is going to be wildly different? Or in the case of intelligence, in which, the social structure of our world is going to be totally different, where there's no prejudices or where the educational system is totally different. Then the way that the genes interact with those environments might be radically different. And that's what you see on the right-hand side of this graph, where you've got the relationship between the gene and the outcome, which is on the y-axis, is very different in environment 3 compared to environment 4. This is a case of gene-environment interaction. And this gene-environment interaction response was inspired by work in the biological sciences that Lewontin was familiar with. This is some data taken from fruit flies, where the x-axis represents environmental differences, so different temperature environments. And you can see that they're not uniform lines going in the same direction. These genotypes, these different lines, they interact. There's different kinds of effects of the environment on that genotype across those different environments. So there's two ways to respond to this in the human case. Some behavior geneticists have hit back saying, well, sure, we can find these things in fruit flies, but the evidence for gene-environment interaction in humans is that it's just not ubiquitous. It just doesn't seem to be there, so let's not worry about it. Or you have other people like philosopher Nevin Sesadek, who thinks that the causal claims can still be made in spite of this interaction. That first response and the initial criticism by Lewontin and others is essentially, I think, a debate about the degree of invariance of a causal relationship. So if you've got data that corresponds to what you're seeing here between E1 and E2, and you expect the way that the gene is expressed to be similar across a whole range of environments, then you're making a claim about the degree of invariance or stability of how that gene is expressed across different kinds of environments. You expect it to be expressed the same way across all sorts of environments. Then you might not think that extrapolating from a heritability estimate in a sample to a more general population is so problematic. You believe that there's going to be stability. If you think there's not stability, if you think there's no invariance or there's a lesser degree of invariance, then you might believe that there could be these gene-environment interactions, that genes will be expressed differently in different kinds of environments, and then we can't make these general causal claims using heritability estimates. So this debate in behavior genetics and the philosophy of biology about how useful heritability estimates are, I think comes down to different perceptions and how invariant these causal relationships that we're discussing when we talk about heritability are. Okay. All right, so just to summarize this then, so the single gene traits, they're characteristically specific and invariant, and this lends themselves to deterministic thinking in some cases at least. However, the heritability debate, these genes, we don't know if they're specific because we're talking about the effects of lots and lots of different genes. And the debate about how useful heritability is as a concept hinges on whether the causal relationships are invariant or not. These are these type causal claims. But what both of these fields are indicating is that when we think about genetic causation, the notion of invariance, which is related to determinism, and specificity do look like they influence genetic causal reasoning. They do look like they make a difference to the kinds of causal explanations we're willing to accept or reject. Okay. I want to move now to talking about a psychological framework for understanding genetic causal reasoning that I think lines up quite nicely with the way that philosophers have identified features of causal relationships. And this is the genetic essentialism framework. So the genetic essentialist framework has been proposed by Ilan Nimrod and colleagues. He's a social psychologist at the University of Sydney. And instead of looking at general features of why we pick out some causes as more important than others, as I've just illustrated, he thinks that the way we reason about genetic causation is rooted in this more general cognitive mechanism of psychological essentialism. So psychological essentialism is a process of representing and categorizing natural entities like organisms, like ourselves. And it's based on the belief that we all have these unique and distinct essences, so some sort of underlying nature, and these essences make us who we are. So according to Dunn and modern colleagues, cultures have attempted to place some sort of concrete construct as the essence placeholders. In the Hindu tradition, this was prana. In traditional Chinese culture, it's qi. In Galenic medicine, it's the balance of the four humours. And then in the modern day, this essence placeholder is encapsulated by the gene. And this is sort of supported by the rhetoric of things being in our DNA or our DNA making us who we are. So he thinks that genes fill this special essence placeholder role, and because of this, we have a particular way that we think about genes when we think about causation. And the way that he's tried to investigate this is he's come up with this genetic essentialism tendency scale. This is a set of 24 items where participants will rate their agreement with each item. And sorry, let's get the item. These are the items. You can see the first two, determinism and specific etiology, match up quite well with the features that philosophers have been talking about as much as explaining causal selection or causal reasoning in a more general sense. So in the genetic context, determinism is the thesis that the effects of a gene are inevitable or immutable. And these are kind of encapsulated by the rhetoric of things, you having a genetic destiny or a genetic curse. Specific etiology is about specific traits arising from the action of single genes or specific genes being the gene for something. Then there's these other two features, which I actually think do correspond to some of the other features that I'm not talking about today, like valence and abnormality. But he also thinks that when people causally reason about genetics, they succumb to the naturalistic fallacy. So if certain genes are thought to make traits natural, and then those traits are moralized about. And also, when you believe in genetic essentialism, you tend to group people into these homogeneous and discrete groups in virtue of what you perceive their shared genetic essence to be. So these kinds of studies, they've used their scale, the GETS, to look at whether the public think about genetics this way, asking them a whole lot of questions about how they think genetic causation works. And they found that most people did correspond to what he calls these biases. They also found that determinism and specific etiology were the most strongly correlated, but that the model in general fit a four-factor model better than a three-factor collapse model. And what? Oh.
One second. There's an
Yep.
announcement, a sort of window
Oh,
we see.
yes,
Okay, thank you.
sorry, I've moved that out of the way. I'm trying to hit snooze. I'm trying not to stress about this while I can
Thank you.
talk.
Thank you. I just
But
wanted to let you know. Thank you.
no, thank you. I'll get that out of the way. It's counting me down. It says I have 22 minutes, which is roughly what the rest of this talk will take. It may mean that I log out and log back in again for question time, so I'm so sorry. It's just not letting me snooze it, so I'm just conscious of that countdown. Sorry, I shouldn't have put it on that screen. Okay, all right, so that's his that's his four-factor model of genetic essentialism, which I've worked with Ilan and others like Paul Griffiths to show that these features of genetic essentialism do have parallels in the more general causal reasoning literature. So hopefully the determinism and the specific etiology links are fairly obvious. Naturalness, I think, can be related to the abnormality of the causes and the effects, and also the way that those are thought about in a moral sense, the moral valence of them. And putting members into homogeneous and discrete groups, that's the way that we perceive the effect of genes that may also correspond to the way that we assess the moral valence of an effect. Okay, the last part of this talk that I want to discuss, and I do have time according to my countdown, so that's okay, is using this framework and how it corresponds to the philosophy to do some empirical work to see, okay, so Ilan and his colleagues have done these psychological surveys where they've given people questionnaires and explicitly asked them, do you think that genetics works in this way? But with colleagues, particularly Ritzart Reimann, who is a philosopher of science at Macquarie University, who's led the study I'm about to present the results from, we've started to look at, okay, well, how is genetics communicated in the media? Are these kinds of biases encoded in the way that genetic information is communicated, in this case, in newspaper text? So this is a preliminary study looking at whether these genetic essentialist biases have come up in print media, and we're hoping to do more work looking at other media types and other kinds of traits as well. Okay, so for this study, this empirical study, we decided to look at genetic explanations of obesity. And a couple of reasons that we were looking at obesity. Firstly, obesity is a pressing health concern in Australia and worldwide, which means it's important to study, and it also means that it gets a lot of media coverage. So when we're trawling through newspapers to look for these genetic explanations, we hope to get a lot of data when it comes to obesity. Obesity is also something that's potentially exacerbated by an essentialistic media discourse. So there have been some really interesting social psychology experiments to show that when people get genetically essentialistic explanations about obesity, like obesity is in your genes, or you have the genes for obesity or not, then that will affect the kinds of nutritional decisions they make. It will affect whether they decide to whether they feel like they're in control of their weight, whether they decide to eat healthy foods or unhealthy foods, be active, etc. So if there is a media discourse about obesity, which is quite deterministic or essentialistic, we think this could be potentially damaging for people's public health. We also chose obesity because while there are some monogenetic or Mendelian diseases which have obesity as a symptom, so you can see in this picture here, the monogenic obesity phenotypes, in these cases, they have a large genetic effect and they correspond to a single gene mutation. They're very specific. Most cases of obesity and most of the discussion we found in these texts about obesity did not correspond to these monogenic traits. They were corresponding to the polygenic, the more quantitative traits, where the effects of genes on obesity are modest. The environment makes a very big difference, and it's lots of very small effects of many, many genes when they contribute to obesity. So the reason we focused in on obesity was because if we did find explanations that were specific or deterministic, then we'd think that they would be biased ways of communicating that media. They wouldn't correspond to our best scientific knowledge about the genetics of obesity. And so what we did, we used the Australian obesity corpus put together by Monica Bednarek at the Charles Perkins Centre, which consisted of over 26,000 newspaper articles about obesity. And this was across 12 major Australian newspapers, so a national newspaper and one or two from each state or territory. And we started off by looking for any of the sentences containing gene-related terms. So we had these gene-related terms that we searched through the text for: gene, genetic, etc., genotype, and that brought up just over 4,000 sentences. And then we started with human coders looking for the absence or presence of these four features from the genetic essentialist framework: determinism, specific etiology, naturalness, and homogeneity. And as we went through coding these according to some of the things from Dan Nimrod and colleagues' works and also what he used in the gets, we started supplementing our coding scoring with coding rules. So I've got some examples here for each one. For instance, we would code something as specific when the name of the trait attributed to the gene was contained within the name of the gene, so such as the fat gene or the skinny gene. We would code something as deterministic if there were some metaphors about hardwiring, like programming or controlling. We would code something as naturalness when genes were invoked to exculpate moral responsibility, invoking the naturalistic fallacy. And homogeneity when genes were invoked to describe kinds or types or tribes of people. So here are a couple of examples of the way that we coded these sentences. So I've got an example for each of these, as well as one that we didn't code as having a bias at all, even though it was a genetic explanation. So you can see for the determinism example, it's talking about being genetically programmed with taste buds that respond to fat, sugar, and salt. In the specific etiology case, we've got the gene for obesity. In the naturalism case, people are talking about things in evolutionary terms and it being more natural to be overweight than to be thin. In the homogeneous and discrete case, the group that's referred to here is sumos, so that sumo wrestlers have a particular kind of genotype and they don't experience the same things, the same complications that other people do. And this last case is one that we didn't code at all for any biases. So it talks about, a pair of people, two sisters, sharing a genetic predisposition not lending itself to any of this sort of deterministic or controlling type language, and that those genes were inherited. That's a relatively unproblematic way of communicating about genetics. Okay, so what we did is we coded all these sentences by hand and we came up with these coding rules. And then with the hand-coded sentences and the rules, we fed these in, and this was largely Ritz who did this work, fed these into 5 different models, 2 machine learning models and 3 large language models. And then we looked at how well these models performed in terms of precision, recall, and F1, where precision and recall roughly correspond to their type 1 and type 2 errors, right? Whether they're capturing all instances of these biases when they're there and, they're not kind of overreaching and capturing things when they shouldn't be. F1 is a balance between precision and recall. That was a sort of an estimate of both. And I'm just showing you on screen the results from our sort of worst performing model, which is this random forest machine learning model, where you can see that in some cases, like with determinism, the recall was very, very low. And our best performing model, which was the GPT 3.5 turbo few shot model, where we had really good precision and recall and quite a nice F1 score there. So, and looking at the way that other models have performed in other kind of classification tasks in the literature, this was within or above those standards. And so once we got these results for GPT 3.5, this model, then we decided to run it on all of the whole corpus to look at the prevalence of specificity, determinism, naturalness, and homogeneity in these newspaper texts. Okay, so this is our data based on that best performing model. So you can see, first of all, if we just look at these descriptive statistics here about what was in the corpus. So as I mentioned, there was just over 26,000 articles talking about genetics in the obesity corpus, which corresponded to 800,000 sentences. Of those, 1,849 mentioned genes, and of those 1,849, 578 contained one or more of these biases. And as you can see here, what we ended up finding the most was sentences and articles. This is at the sentence level, this data, which described the genetic causation or genetic explanations that were deterministic or that had this sort of gene for rhetoric lending itself to specific etiology. We found much fewer sentences relating to naturalness and a few more when it came to homogeneity. One other thing we looked at is the co-occurrence between these features. So if you remember with Alan's work, he found there was a strong correlation between specificity and determinism in his participants. So we looked at which of these co-occurred at the sentence level and at the article level. And what we found was the co-occurrences at the article level and the sentence level were both very similar. At both levels, specificity and homogeneity was actually the most frequent co-occurrence, followed by specificity and determinism, which was this one we would have expected given the genetic essentialist framework. And it's quite interesting that these patterns of co-occurrence are occurring both at the sentence and at the article level. So there seems to be something about when people do invoke, we didn't have as many data points about homogeneity, but when people were talking about these homogeneous groups, they were also talking about specific genes. And then we had this correspondence between specificity and determinism also. The last thing we did is we had in mind that we might name and shame particular media outlets. We didn't go as far as to doing this by author, but we looked at the different media outlets to see, whether some newspapers were more biased than others when they were communicating about genetics. And at first glance, when you look at this, it looks like there actually was quite a lot of variation. This is the total number of biases we found in each of these newspaper outlets. The Sydney Morning Herald, Sydney being my home city, performed the worst if we're looking just at total number of biases per outlet. And in fact, 1/3 of the vendors contributed more than the remaining 2/3 combined when we were just counting up these biases in this sort of straightforward way. However, what we did have to check is, was there variation amongst outlets in how often they were talking about genetics in the first place, right? In the number of gene sentences that were actually occurring in this corpus when we got this representation from newspapers. And as you can see, where the Sydney Morning Herald did have the most number of biases for all these newspapers, it also had many, many more articles and sentences about genes. So what we really need to just look at is how many biases per gene sentences or per gene article were there. And when we adjusted for this, we found that this variation in how many biases there were per outlet largely disappeared. You can still see the Brisbane Times is actually sticking out a little bit more than the others now. You can see that there's not so many differences when we control for that fact. Okay. All right, so in conclusion, in general, when people causally reason, they tend to pick out causes that are more specific, more invariant, and all these other features I didn't get to talk about, intervenable, abnormal, negatively balanced, and involve agents. All of these things tend to make a difference to the psychological way that we causally reason, that we pick out important causes. In the genetics case, genes involved in Mendelian or monogenetic traits quite often are specific and invariant. However, in the polygenic context, this is often not the case. It's not specific, and there's quite a lot of debate about their invariance. However, the genetic essentialist framework suggests that at least the public tend to think of genetic causation as specific, as deterministic, as well as natural and conferring homogeneous group membership, which perhaps suggests that people have this sort of way of thinking about genetics that best corresponds to monogenetic traits. I know this is very much in line with Greg Raddick's work, which have these features of specificity and invariance. And our corpus analysis study suggests that this way of thinking is present in the print media about obesity at least in the Australian context in newspapers, and it's particularly specificity and determinism that come up. And this is our first study using these methods, but what we'd really like to do is expand this research to look at how genetic causal reasoning bears out using these methods in other contexts, by looking at other phenotypes like sexuality, intelligence, personality, as well as a whole host of health and disease states. We also want to look beyond newspapers to look at transcripts from radio, television, podcasts to see if people communicate about genetics differently in those contexts, to also look at social media. This is something Ritz has done in other contexts. And finally, to look at academic print. Academics tend to think we're immune from these kind of causal reasoning influences, but studies in other contexts suggest that we're not quite as immune as we think we are. And then finally, something that I plan to do with other colleagues here at the University of Melbourne and at Monash is to look at whether the causal reasoning frameworks in genetics have some correspondence for other biological factors, such as the way people causally reason about microbes and about the effects of gender and sex. So that's it. Big thank you to all of my colleagues and collaborators, but especially Ritz, who has, as I said, conducted the bulk of that work on corpus analysis. Thank you.
Thank you very much, Kate. Um, do you think you must reconnect, or we can start the Q&As without any?
I'm getting I still have this message that says I've got 6 minutes. So I've it's my fault. I've been pressing snooze on this thing all day and now it's not letting me press snooze anymore. I should have restarted it earlier. So I'm so sorry. Do you mind if I let it do its thing and then I'll be back in a minute or two?
No, that's perfect. Just do that so that we can have the time we need for discussion. So yeah, please do that.
Okay, thank you. I'll see you in a moment.
So just to let everyone know, I'm interrupting the. No problem. So recording starts, resumes. So we
Yep.
have the first question by Bertrand Daignan-Fournier. Please just introduce yourself in a few seconds so that Kate knows who you are.
So, just as Thomas said, I'm Bertrand Daignan-Fournier, and I'm a yeast geneticist. Um, so, thank you very much for the talk. I was wondering, in the cases where a single gene explanation for a trait is not sufficient, people tend to use polygenic explanation rather than think in terms of pleiotropy or epistasy and this kind of explanation. And there is this issue of missing heredity
Hmm.
that, well, make us think that polygenic additivity is not maybe the right way to think about it. And maybe thinking in terms of alleles rather than genes would be more accurate. What do you think about that?
Yeah, I mean, the point about thinking about genes rather than alleles is a good one because if we're talking about the public perception and public genetic causation, there's probably not an understanding or a distinction of what's going on there. So would your suggestion be when there's not strong invariance or specificity to not go straight to the quantitative route, but to try to look for, I guess it just depends on the tractability about how many other modifiers there are in the system. I do take your point though that, and this is something I didn't talk about in the talk, is that these heritability estimates, because of the problem of missing heritability, are generally thought to be an upper estimate, and that there's lots of literature out there thinking that they might be conflated with they might be inflated by environmental variance that's just confounded or correlated with the genetic variance. And that's something that I do believe actually is that these heritability estimates can be inflated by gene environment correlations.
Second question by Rebecca.
Hello, Kate knows who I am, but
Yeah,
for everyone else, Rebecca, I'm a PhD student at the University of Sydney. Um, I was curious, Kate, thank you for the talk, by the way, that was great. It kind of links to some discussions I've been having recently, and I'm curious how the kind of claims that you can use causal language for the genome-wide association studies, because they kind of fit in a different category to the heritability studies. And I'm curious about how you think it fits in your framework.
right. So how I guess what I'm mostly doing here is looking at how people interpret it rather than how people should interpret it, which is a whole other huge area. And particularly with the GWAS stuff, people like Lucas Matthews have done quite a lot of work trying to figure out exactly what these causal relationships are. When it's about how people do interpret these results, again, at least in the public setting. So all of the stuff we looked at in that obesity corpus, we didn't have time to go through every sentence, but it'll be a mix of heritability studies, of GWAS studies, of, in some cases there might be these monogenic Mendelian cases, but the sample we looked at didn't have any. But it's completely abstracted when it comes to the public communication. So it will just be gene for, I guess, would be those sort of Mendelian thinking, or the genetics of, or mostly genetic, that kind of thing. And again, it's hard to know how much the public have an awareness of these different ways of estimating genetic influences. I'd say that probably most members of the public don't think that, or don't know that there's this whole range of methods in genetics that correspond to quite different kinds of causal claims that you can get out of them. So that's an additional complicating factor.
Um, Robert.
Hello, thank you for the interesting talk. Um, so my question is, um, so in the philosophical part, it seems you said that causes are selected for specificity and invariance, and from this perspective, it kind of sounds like these are the properties of causes that we can have some criteria for. But then if you look at it from the psychological side, then it sounds a bit more like, people can construe any cause as specific and invariant due to their kind of essentialism, and it's kind of
Mm.
cultural dependent on what they choose. They could just, I mean, what they choose as this kind of essentialist placeholder could be anything, could also be some kind of environmental factor in a way. So that makes it kind of sound very culturally relative. And then basically any cause could be construed as selectable because it will be construed due to this psychological essentialism as specific and invariant. But then if we are kind of like, if we think there are criteria for specificity and invariance, and if we are realist about these properties of causes, then we would think that the biases should actually be biased towards kind of the real causes, and this kind of psychological construction of essentialism should not be entirely free, free in a way.
Yeah. I mean, I think there's some plausibility that, the reason that genes fill this essence placeholder role is that they are perceived to have these kinds of properties that are maybe good reasons for picking out causes. So we don't tend to use the same kind of language saying, our childhoods make us who we are, or our diets make us who we are, or these other kinds of things that have a causal influence, but they're not specific or invariant in that same kind of way. I think the problem in the genetic context is people, there are some contexts where genes are specific and invariant, and so it does make sense to sort of point them out as important causes. But because there's this range of genetic methods, as I was just talking to Rebecca about, and ranges of ways of making causal claims in genetics that don't always correspond to specificity and invariance, there's still this perception that all genetic relationships might have specificity and invariance, whether it's implicit or explicit. Um, yeah, so I think the specificity and variance, I do think they are good reasons to pick out causes, and it might be why genes are in that do fit that essence placeholder role.
Thank you.
Kate, I have a question about heritability. What is the evidence for arguing against Lewontin that it's okay for Drosophila to say that there is this very strong dependency on the environment, but you seem to suggest that some people say it doesn't apply that much to humans. And I just wanted to know a little bit more about the arguments or evidence for that.
Yeah, so they are empirical responses. Those two references I had there are behavior geneticists who have said that most of the it depends on the field of genetics as well. But in behavior genetics, if we're looking at something like IQ or aggression, that we're getting the same kind of heritability no matter which population. So we've studied lots of different populations. There's lots of studies out there. We're getting a similar kind of heritability estimate. Therefore, even in a population that we haven't done the heritability estimate for, it's reasonable to extrapolate that. In the same way as if you were doing a drug trial and you do 20 different studies across the world and you get a similar kind of efficacy, then you've got a sort of good reason to extrapolate it over to this other population. And that's sort of what Sesadek is saying in his response as well. He wants to say something a bit stronger about type causal claims, but that you don't have to have this kind of law-like invariance that I think Lewontin wants, where this relationship has to hold not only over the populations right now, but the future populations and the possible background conditions. But, we can make some sort of general causal claims based on the evidence we have from what's in front of us. That's sort of the tension. It's about invariance, but it's also about the relevant background conditions, right? Do we need to think about these hypothetical ones, or can we just be thinking about the kinds of relevant ones that we're measuring?
Okay, thank you. Very useful. Bertrand.
Yes, it seems to me that some of the biases you described are linked to a view of what genes do. And this comes back to a historical description of genes by Biddle Tatum in the 1950s, where they claim one gene and one enzyme, and beside that, one major function and one trait, which was auxotrophy for vitamin in filamentous fungi. And it seems to me that this is still the way many people are thinking about what genes do. And for this reason, maybe redefining genes could be a better way to think about that than, well, I don't know. But, well, let's say, do you think that this bias is still very present, and how can we deal with that to get it less present?
Yeah, it's a great question. So, I mean, I do think they are, based on our studies, at least, and the other kind of psychological evidence, it does look like it's present, at least in the public. It's an open question as to whether it's present in professionals and people who have been educated in genetics. So I mentioned a few times Gregory Radic, who does take the line that it is contingent on the history of genetics, that people tend to think in this Mendelian fashion, but also there's something in our education system where people who get some, when they do get some exposure to genetics, they're doing Punnett squares and they're learning about these very simple one gene, one trait type relationships. And he's actually done some experimental work. So if we're thinking about what interventions we can make so that people aren't succumbing to these biases, he didn't measure these kinds of biases explicitly in the way we have, but he's done some experimental work that suggests that if you change the way you educate people about genetics, if you don't start with the Punnett squares and thinking about these Mendelian traits, and if you educate people about the complexity of genetics and the different way that genetic traits can develop, then people are going to be less deterministic and less likely to think like that. So it would actually be interesting to do some stuff in combination with those educational interventions and then measure things like the genetic essentialist tendency scale.
Thank you. Parvati.
Hi, am I audible?
Yeah.
Okay, hi. First of all, thank you so much. It was a very interesting talk. I wanted to ask, how do you think the current discourse on epigenetics, for instance, impact people's views on genetic determinism and other biases?
Yeah, that's a really good question. I feel like this is I can have my kind of anecdotal and my intuitions, but I feel like that's actually a very fruitful empirical question, where you could, if you were to do, say, the psychological studies where you give people questionnaires, you could also include some questions in there about, have they heard of epigenetics? What do they think epigenetics is? And if they, have this concept of things modifying gene expression, whether that modifies how essentialistic they are. That's a really, really cool suggestion. I would worry that people would start to think of epigenetics in the same, it's just a different layer of, they're specific and they're determined in a different kind of way. But it's not something I've done any research on. I think it's a really fruitful area for future research.
Thank you.
Next question by Lucia.
Hi, everyone. So I'm Lucia. I work at UWA, so Western Australia. So I'm on the other side of the country where Kate is. So what I was wondering here is just a curious question, actually. When you were doing the studies on the different newspapers and outlets, if there was pushback on the essentialism, because what I see a lot talking about genetics or determinism with lots of people coming from biology as well, and now they work with social sciences. So there's a lot of pushback on that determinism, right? That we actually have so much that is only cultural, or that we shouldn't be kind of focused on genetics. And you can see a lot on the public, this idea that, we can kind of disconnect from our nature in some kind of way. So I was wondering if news about genetic work, right, or thinking about genes or how that affect who we are or our behavior and all that, kind of also comes with a pushback on the essentialism or the potential essentialism of that of those genes.
So yeah, one thing, so we didn't get to read every single sentence and article. The reason we did the machine learning was so we could do it at scale. But when we were coding them, we had quite a long list of rules for excluding things. And one of the things we did exclude were things like, if the gene worked in this way, if the gene was like this, then we would get this, right? So it was thinking hypothetically about if there was a gene for fatness, then it would be this, but it wasn't making the concrete claim that there is a gene for this or that genes are deterministic. And some of those sentences probably had features where it might have said, most people think genetics is determined, but actually our best science tells us not, right? So it is exactly what you would say, going in that other direction. All we did in that study was exclude those because we didn't want to obviously code those sentences as being deterministic just because they were mentioning determinism. They weren't actually proponents of determinism. But we didn't go through the rest of those sentences and look at whether they were pushing back in the other direction. We didn't measure that at all. That's a really interesting suggestion, and I'm just trying to think of what would be the coding rules? Like, how would we identify the anti-essentialists or the anti-determinists? We could probably come up with rules in a dictionary to get that kind of information. I mean, another thing we want to do in follow-up research, actually, another thing we didn't look at was in that same corpus, how many environmental explanations were there? So we looked at, all the gene sentences, gene, DNA, genetic, genome, etc. And initially we wanted to do a parallel where we looked at, okay, if there's X amount of genetic explanations, let's see how many environmental explanations, where it talks about diets leading to obesity or lifestyle leading to obesity or, childhoods or whatever it is. But it just became a bit intractable to find a equivalent way of conceptualizing the environment and picking out the terms for environment, because environment, with the genes, you've just got a few terms that will tell you about genetic explanations. With environmental explanations, it could just be so many different things: lifestyle, environment, upbringing, diet, exercise, etc. And so that we didn't do that part of the study, but it's certainly important because at the moment all we're talking about is there are some claims in there that don't seem justified, that they do seem biased and they're deterministic and essentialistic. But of course, that could be balanced out quite well with some much more nuanced way of describing genetic causation. If you can think of a way that we could measure this, if you could think of some of the terms we could use, please let me know. Yeah, definitely. Yeah, we'll have a chat.
Now a question by the most Australian member of our team, Jonathan.
Nice. So yes, hopefully you know who I am, Kate, and nice to see you again.
Honorary Australian.
Indeed. Thank you very much for a very nice talk. So I imagine you get this question a lot, perhaps in this context. So I'm curious to see your thoughts. Given that the kind of presence or the prevalence of bias in these different areas, either in the media or public understanding, I mean, what can we do about this in a sense, and to kind of address this bias, to kind of reduce these biases or change these biases in some ways?
Yeah, it's a really good question. So, I mean, one suggestion is that intervening on the education system, that potentially if we learn about the complexity of genetics, then people will be less likely to succumb to these biases. That's probably just going to be one small intervention. Another really open question is if media outlets change the way they communicate about genetics so that they're not communicating, that things are programmed or controlled or that there's a gene for, whether that's going to kind of infiltrate the public consciousness. And say we improve the way we communicate in the media, then you've done, and Rod and colleagues do another study in 20 years' time, they might find that due to the changed media landscape, people are scoring lower on the gets. That's sort of one hypothesis, that the way we communicate in the media could influence people's genetic essentialist tendencies. This is a little bit at odds, though, with thinking that genetic essentialism is this psychological mechanism of essentialism and that we have genes as the essence placeholder. So it depends a bit on whether you think genes as an essence placeholder is kind of deeply rooted, that it's deeply rooted in our psychology, that we need to find some sort of essence placeholder and the genes are the best thing. And if we move away from the genes, maybe it'll be epigenetics, right? People are going to latch onto something else. Or whether it's or whether you don't buy that part of the story that it's a deeply rooted psychological mechanism and you think instead that it's a more culturally constructed phenomena, in which case you might think that intervening on the media landscape is going to be really effective. If we could just communicate better, if the journalists can work more closely with the scientists and be more careful about the claims they make, then people will be less likely to succumb to these psychological tendencies. At the moment, I think it's, because we're all kind of entrenched in our culture, I think it's almost impossible to tell which of these are having an influence. Maybe there's some cross-cultural studies you can do where the media landscape is very different, and you could see if people have different kinds of essentialistic tendencies. But if you've got any suggestions, if you know of any places where you think the media might be quite different, yeah, that would be a really great place to start.
I don't see any other questions. I think we had a wonderful discussion. If there is any other question or comment, please manifest yourself. No, it seems that that was really great, Kate. Really interesting, and I really enjoyed the discussion as well. Oh, Reen.
In fact, Rin is the expert on on Jonathan's questions. So, hello, nice to see you.
Sorry for taking my time. I couldn't find my raise hand thing. Hi, Kate. Yeah, so I'm Reen. I'm beginning a postdoctoral position at the University of Barcelona soon. So I had a question about the first part of your talk, and it's going to be a little conceptual clarification question. So you distinguished between like the two cases, single gene trait cases and quantitative cases, and you distinguish between them in terms of whether or not it's legitimate to draw inferences about token causation from claims about type causation, which genetic studies directly support. And you had this table to describe the difference. And I thought that your description of the two cases was asymmetrical. So when you describe the quantitative case, what you say is, you describe the type claims as tongue rolling is mostly genetic, and you describe the token case as my tongue rolling is mostly due to my genes, and you say that it's not okay to conclude the latter from the former. Yeah. But what you say about the single gene case is that, well, you describe the type claim as saying X gene causes tongue rolling, and you describe the token case as saying X gene causes my tongue rolling. And you have no reference there to, let's say, the proportion of the size of the effect that the gene is said to cause. And I was wondering, why is the difference there? Is it implicitly assumed that also in single gene cases, the type claim is actually a claim about gene X causing most of the variation, and the inference to the token case is legitimate because in these cases we can indeed infer that also the trait of a particular individual was mostly or all of it was caused by the gene. But that doesn't sound very, I don't know, plausible to me, because I mean, even in these single gene cases, like what does it even mean for one gene to cause all of my traits? And the only like ways to conceptually unpack it would also render the inference in the quantitative cases from type causation to token causation actually doable.
So the reason, I'll start with the single gene case. The reason I think that you can go in a lot of the single gene cases from the type to the token is it's to do with this invariance, and it's to do with the way that people counterfactually reason. So if you've got something that's highly specific and highly invariant, so you've got, say, there's just one gene that if you have the mutation, you can roll your tongue, and if you don't have the mutation, you're not able to roll your tongue. And so at a population level, it fully explains the variance in tongue rolling. So it's the difference maker to tongue rolling. It doesn't matter about anything else in people. This is an abstracted case. But having that gene means you can roll your tongue, and not having that gene means you can't roll your tongue. So at the population level, we can say that it's the difference maker. It always corresponds the gene and the tongue rolling. Then at the individual level, you can make a sort of counterfactual claim based on that difference making claim at the population level, where I have the gene for tongue rolling, but if I did not have the gene, then I wouldn't be able to roll my tongue. And it's really clear because it's just this single gene specific invariant case what that counterfactual would be. If I did not have it, then it's inevitable that I wouldn't be able to roll my tongue. And that only works if something's sort of very specific and invariant like that. The heritability case, heritability is a very weird way of thinking about genetic causation. So you can also think of it as explaining variation at the population level in the same way the gene for tongue rolling might explain variation at the population level, where variation is just can roll or cannot. But with heritability, the variation at the population level is on this spectrum, whether it's height or intelligence or whatever. And we're not identifying any particular genes as mapping to that variation. We're just identifying what we think is genetic based on inferences about familial resemblances, say, in the case of twin studies. And so you can perhaps make a causal claim or give some sort of explanation about, okay, the variance in the heritability case, the variation in height can be explained by variation in genes. Now, there's lots of empirical kind of reasons to push back on that. As Bertrand already mentioned, this might just be an upper estimate. There might be other confounding environmental factors in there, but you still, I guess, are explaining what causes the differences at the population level. But at the individual level, there's so many different gene combinations that, if you were to think about an individual causal claim, if I did not have the genes I have, then I wouldn't be 165 centimeters tall. It's not clear what that counterfactual is. Which genes do I think about being different, right? What would not being 165 centimeters tall look like? Would I be 175 centimeters? Would I be 150 centimeters, etc.? Because there's so many different values of the cause and the effect. So I think the reason the single gene case works is because there's only a single value of cause and single value of the effect, and so the counterfactuals are really clear, and that's why you can go from type to token. In the heritability case, it's so complex that it doesn't make sense to kind of causally reason in the individual sense.
Okay, thanks.
All right, thank you very much. There was a question in the chat about the availability of the recording, and the answer is yes, the recording will be available presumably tonight on the Fit in Biomed website. Kate, thank you so much. That was great. I just want to say that our next seminar will be on November 21st, I think, with Andreas Wagner. And we will talk about, I think, robustness. I don't have the title yet, but robustness is a likely candidate for that talk. And that's going to be in person, which has become relatively rare. So if you're around Bordeaux, please come to talk with Andreas. Thank you so much, Kate, and see you, I hope, in the near future.
Yeah, thank you so much, everyone. Thanks for the great discussion.
Thanks. Bye-bye, everyone.
Bye-bye.